In the context of higher organisms such as mammals, semen is defined as the viscous biological fluid produced from the reproductive organ of reproductively mature male members of a species during the act of ejaculation, which usually occurs at the culmination of sexual intercourse or masturbation. Semen consists of two main parts: the spermatozoa or sperm cells, which comprises of about 5% of semen, and the seminal plasma or seminal fluid. The seminal plasma consists of secretions from the seminal vesicles, the prostate, and the bulbourethral glands. Among the many sugars, amino acids, enzymes, and minerals present in the seminal fluid, the most noteworthy and major energy source for sperm cells is fructose. Secretions of the seminal vesicles contain fructose, along with substances that suppresses any immune response against the foreign sperms inside the female reproductive tract. Secretion of the prostate gland contains substances that help to stabilize and protect the genetic material and thereby the fertility of the sperm cells. Secretion of the bulbourethral glands, also known as Cooper's glands, contains substances that provide viscosity and slimy texture to the semen, thereby increasing sperm motility within the female reproductive tract. It is also the source of the pre-ejaculate. Semen analysis or semenogram or spermiogram is a comprehensive series of clinical tests that evaluates the quality and quantity of a semen sample. The most important and commonly analyzed parameters included under semen analysis are liquefaction time, amount, color, pH, odor, viscosity, sperm motility, sperm viability, sperm morphology, sperm count, and fructose content. In this video, we'll be performing semen analysis of human semen based on the previously mentioned parameters. For this experiment, you will need the following: freshly collected human semen sample, 10 ml graduated cylinder or syringe, pH paper, preferably narrow range, a narrow glass tube or pipette, droppers, glass slides and cover slips, eosin and nigrosin stain solutions, Harris hematoxylin stain, eosin stain, 1% acid alcohol solution, alkaline water or 0.2% ammonia water, 95% ethanol, absolute ethanol, xylene, DPX or Canada balsam, Coplin jars, thumb forceps and pins, hemocytometer counting chamber with WBC pipette. Semen diluting fluid, compound light microscope, immersion oil, Seliwanoff's reagent, test tube and holder, spirit lamp, 5 ml and 1 ml measuring pipettes. The first and foremost parameter in semen analysis is the liquefaction time. Liquefaction of semen is the process that results in the conversion of freshly ejaculated semen from a thick, whitish, opaque, viscous, and gelatinous consistency to a watery, less viscous, translucent fluid with the help of proteolytic enzymes present in the seminal fluid. Liquefaction time is the time taken for this conversion process to occur. To perform this test, note down the time when the semen sample was collected. Observe the semen sample and note down the time when the liquefaction is complete. The normal liquefaction time for human semen is 10 to 30 minutes. Any period of time longer than 30 minutes is indicative of infection. Before estimating semen volume, the semen sample needs to go through the liquefaction period. Allow 10 to 30 minutes after collection for the sample to liquefy before measuring the volume. To determine the volume of the semen sample, tilt the sample container for a couple of minutes and then carefully pour the liquefied semen sample into a 10 ml graduated cylinder completely till the last drop. Wait a couple of minutes for the sample along the inner walls of the cylinder to settle down, and note down the volume. Add a volume of 0.5 ml to the observed volume in order to compensate for the semen sample stuck to the walls of the collection container. An alternative method of measuring semen volume is to directly aspirate the liquefied semen sample into a 10 cc needleless syringe. After positioning the container in a slanting position for a couple of minutes prior to aspiration, make sure to aspirate completely till the last drop, while at the same time taking extreme care not to aspirate any air bubbles. Note down the volume as before. Add a volume of 0.5 ml to the observed volume in order to compensate for the semen sample stuck to the walls of the collection container. A third method, which is also considered the most accurate method according to some published papers, is to directly weigh the sample in an analytical weighing scale. 1 gram is generally agreed to correspond to 1 ml of the sample. This method eliminates the loss in volume when transferring the sample from the collection container into the measuring receptacle. One important point to keep in mind when applying this method is to note down the weight of the empty container prior to sample collection. Approximately 2 to 5 ml is considered as the normal volume of semen per ejaculation. Low semen volume, also known as hypospermia, could be an indication of partial or complete blockage in the reproductive tract, especially the seminal vesicles. It could also be the result of certain infections such as STDs. High semen volume, also called hyperspermia, can be caused by extended periods of sexual arousal and stimulation, abstinence from sex and masturbation, excess intake of fluids, etc. The normal color of human semen is white to grayish-white. Semen color is influenced by diet, age, medication, and certain underlying medical conditions. Red to brownish semen color indicates the presence of blood, a condition called hematospermia. This could be a result of infection in the reproductive tract. Take a strip of either broad-range or narrow-range pH paper and touch one surface onto the semen sample. Observe the pH paper for color change. A more accurate laboratory pH meter can also be used to determine the pH. Normal semen has a slightly alkaline pH ranging from approximately 7.2 to 8. Acidic semen could be indicative of a blockage in one or both seminal vesicles. High alkaline pH could be a result of infection. Normal semen has a characteristic bleach or musky smell due to the presence of tiny amounts of basic amines like cadaverine, putrescine, spermine, and spermidine. Slight variations to this smell could result from a person's diet, including certain medications. The viscosity of semen is determined on a sample that had already been through liquefaction. When normal semen is drawn in a narrow tube or dropper and allowed to dispense from the tube by the force of gravity, it flows out of the tube in the form of medium-sized discrete drops without any trailing strands. Abnormally viscous semen, however, flows out of the tube in the form of long and thick viscous strands that are more than 2 centimeters. This condition is called semen hyperviscosity or SHV and is an indication of inadequate functioning of the prostate or seminal vesicles and also infection. Hyperviscous semen can severely impede sperm motility and can lead to male infertility. Hypoviscous semen or watery semen, on the other hand, could be due to excessive masturbation, ejaculation after an extended period of stimulation, and arousal leading to a low sperm count, and also due to certain underlying infections. The sperm motility test is done within 60 minutes of sample collection. Sperm motility is the ability of a sperm cell to move in the surrounding fluid medium. A drop of liquefied semen sample is taken in a glass slide and gently covered with a cover slip. The slide is immediately observed under 40x objective of the microscope. 100 or 200 random sperm cells are briefly observed one by one to check for motility or the lack thereof. To minimize the chances of repeated assessment of the same sperm cell for motility, the microscopic field of view is changed frequently in a specific order of movement, such as from the left corner of the slide to the right corner, etc., until 100 or 200 cells have been assessed. Based on the extent of motility, human sperms are divided into 4 grades, also known as motility grades. The various grades of motility are calculated in terms of percentage by counting 100 or 200 random sperm cells. Grade A or motility 4 sperm cells have rapid progressive motility. These are the strongest and swim fast in a straight line. Grade B or motility 3. These sperm cells have a slow or sluggish or non-linear progressive motility. These also move forward but tend to travel in a curved or crooked motion. Grade C or motility 2 or non-progressive motility. They move their tails but do not move forward. Grade D or motility 1 or immotility. These are immotile and fail to move at all. The immotile sperms may either be dead or alive, which will be ascertained in the next test, which is known as sperm viability test. In this video taken through the microscope, you can see sperm cells exhibiting different motility grades. Observe the individual sperms followed by the cursor. Normal semen contains at least 50% grade A plus B sperm cells combined. Low sperm motility of below 50%, also known as asthenospermia, can be a symptom of disorders related to testicular function and male accessory sex glands. It could also be a sign of infection or injury in the male reproductive tract. In cases of reduced sperm motility, the semen sample could also be checked for sperm viability. In the sperm motility test, if non-motile sperms are found, one has to assess what fraction of these non-motile sperms are still alive and what fraction are dead. Sperm viability test is therefore used to determine if non-motile sperms are alive or dead. This particular test is applicable in cases where the motility of the sperms are drastically low, that is less than 20%. Viability testing is based on the principle of differential staining property of live and dead sperms, also known as dye exclusion assays. Dye exclusion assays rely on the ability of live sperms to resist absorption of certain dyes, whereas these dyes penetrate and stain non-viable or dead sperms. A combination of eosin and nigrosin stains is commonly used in sperm viability testing. Make sure to watch my video on how to prepare eosin and nigrosin stain for sperm viability testing by clicking on the link given on the top right corner of the screen right now, or the link given in the video description below. Just as in the sperm motility test, sperm viability test is also assessed within 60 minutes of collection of the sample. A drop of liquefied semen sample is taken on a glass slide. 5 drops of eosin nigrosin stain, or 2 drops of 1% eosin and 3 drops of 10% nigrosin stain is added to the sample. The stain and the sample are gently mixed on the slide and allowed to stand for 10 to 20 seconds. Take a second slide, touch one edge of the slide on the sample and prepare a smear of the sample onto a third slide. Allow the smear to air dry. Observe the slide under high power 40x or 100x of the microscope. Dead sperms, that is, the sperm cells that were already dead in the fresh semen sample, are stained pink in their head region, while live sperms, that is, the sperm cells that were motile or immotile but alive in the fresh semen sample, will remain unstained. Observe 100 or 200 random sperm cells and calculate the fraction of live and dead sperms in percentage. Normal semen contains more than 60% immotile but viable sperms. Viable sperms below 60% can be caused by excessive heat exposure of the testes, exposure to certain substances that are toxic to sperms, certain infections, etc. Necrospermia is a condition where there are no viable or living sperm cells in semen. Sperm morphology assessment looks at the proportion of sperm cells with abnormal shape or morphology against normal cells. Defects in the morphology of a sperm cell, also known as teratospermia, can be classified as head defects, neck and midpiece defects, tail defects, and cytoplasmic droplets. The assessment of sperm morphology is done by microscopic observation of a stained smear of the semen sample. Different staining techniques can be employed. In this video, we'll be using the hematoxylin eosin or H&E staining technique, which is simple, quick, and reliable. For this experiment, you will need the following: freshly collected human semen sample, droppers, glass slides and cover slips, Harris hematoxylin stain, eosin stain, 1% acid alcohol solution, alkaline water or 0.2% ammonia water, 95% ethanol, absolute ethanol, xylene, DPX or Canada balsam, Coplin jars, thumb forceps and pins, compound light microscope, immersion oil. H&E staining is performed as follows. Take a small drop of the liquefied semen sample on a glass slide and make a smear using a second glass slide as shown here. Allow the smear to air dry at room temperature. Immerse the slide in Harris hematoxylin stain for about 20 minutes. Remove the slide and give it 2 to 3 quick dips in 1% acid alcohol solution. Rinse the slide with slightly alkaline water or with 0.2% ammonia water. The stained smear now becomes bluish. Transfer the slide into eosin stain solution and leave it for 2 to 3 minutes. Give the slide 3 to 5 dips in 95% ethanol, followed by another 3 to 5 dips in absolute ethanol with 2 changes, which is then followed by 3 to 5 dips in xylene with 2 changes. Add a drop of mounting medium such as DPX or Canada balsam to the dehydrated and cleared smear. Cover it with cover slip. Allow the slide to air dry overnight and observe under oil immersion objective of the microscope. Study the morphology of 100 to 200 individual sperm cells and assess the morphological defects of the various parts of the sperm cells. Calculate the fraction of abnormal sperm cells in terms of percentage. A semen sample that contains more than 30% of morphologically normal sperm cells is considered normal semen. Abnormal sperm morphology can be caused by a number of factors. Sperm count is the number of sperm cells found in 1 ml of semen. Total sperm count is the total number of sperm cells found in the total volume of semen obtained in a single ejaculation event, that is, sperm count multiplied by semen volume. In this video, we'll be using the hemocytometer with WBC pipette to perform the sperm count. For this experiment, you will need the following: freshly collected human semen sample, thumb forceps and pins, hemocytometer counting chamber with WBC pipette, semen diluting fluid, compound light microscope. First, prepare the counting chamber by placing the cover slip in position. Keep this aside. Now draw liquefied semen sample till the 0.5 mark of the WBC pipette. Then draw the semen diluting solution until the 11 mark on the pipette. Gently wipe off excess fluid from the sides of the pipette tip. Now place the loaded pipette in a horizontal position and gently mix the contents for a minute or so by rotating the pipette between the palms of the hands. Discard the first 2 to 3 drops from the pipette. Load the counting chamber with the diluted semen sample. Place the loaded chamber under the microscope and let it rest for a minute or so. Under 40x objective, begin counting the sperm cells within the 4 corners and the central small squares of the central large square. Remember to apply the thumb rule of cell counting by excluding cells found along the bottom and left borders of the squares. You can also check out my video on the hemocytometer. Click on the link in the video screen and it will take you there. This is how sperm cells will appear within the small squares of the counting chamber. Repeat the counting process in the second counting grid of the counting chamber as well. Take at least 2 readings for both counting grids and take the average of the readings. Calculate the sperm count per ml using the formula sperm cells per ml equals number of sperm cells into 20 into 1000 by 0.02. Or in short, sperm cells per ml equals sperm counted multiplied by 1 million. Total sperm count equals sperm count per ml multiplied by ml of semen sample or ejaculate. This formula is used when counting is done in the 5 small squares of the central large square. WBC pipette having 1:20 dilution mark is used. Sperm count above 15 million cells per ml of semen is considered normal. Sperm count below 15 million cells per ml, also known as oligospermia, is one of the leading causes of male infertility. Oligospermia can have a variety of causes such as infection and other medical conditions in the reproductive tract or in the testes, chemo and radiotherapy, varicocele, STDs, vasectomy, frequent overheating of testicles, occupational exposure to chemicals and radiation, drugs, alcohol, or tobacco use, anxiety, depression, and other mental issues. Fructose is produced in the seminal vesicles and constitutes about 99% of the reducing sugars present in semen. It is the main source of energy for the sperm cells. Semen samples normally contain fructose and can be detected by qualitative analysis using Seliwanov's reagent. Quantitative colorimetric estimation can also be performed to find out the exact amount of fructose present in the sample. Fructose test is an optional parameter in routine semen analysis and is performed especially when the semen sample contains no sperm cells, a condition known as azoospermia, or when ejaculate volume is below normal. Qualitative fructose test is performed using Seliwanov's reagent. The test is based on the principle that when fructose is heated with resorcinol in an acidic medium, fructose is converted to hydroxymethylfurfuryl, which then condenses with resorcinol to form a red precipitate. To perform qualitative tests for fructose in a semen sample, take 5 ml of Seliwanov's reagent in a test tube. Add 0.5 ml of the semen sample to the tube and boil the mixture for a minute or so. The formation of a red color in the tube is considered as positive test for fructose. No change in color indicates an absence of fructose in the sample. To know the exact amount of fructose present in the sample, quantitative fructose tests may be further performed in a colorimeter using standard fructose and resorcinol reagent. Semen fructose content of 200 to 400 mg per deciliter is considered normal. Low or no fructose levels in semen is indicative of conditions such as testosterone deficiency, obstruction, complete absence or developmental defects of seminal vesicles, and/or vas deferens. So this is all about the semen analysis as performed routinely in a clinical lab. If you found this video helpful, please do show your support and like, subscribe, and comment. Thanks for watching. Stay tuned for more.